What's up, Sci-Fan? Welcome back for Steps of Mitosis, Part 2. Now, in the second steps of mitosis, we call it the steps of mitosis because it is like a dance. We're stepping along one step at a time, and you got to start at the beginning and to end at the finish. And to go through all the steps at the right order is the full dance. So here we are learning about the dance of mitosis. So in part 1, we looked at interphase and we learned how the organelles in DNA are duplicated. Those centrosomes also are duplicated and they start to go to opposite ends of the poles. In prophase, they form those spindle fibers between those centrosomes and the DNA chromatin starts to condense. Then the nuclear membrane dissolves as those chromosomes fully form and the spindle fibers attach to the chromosomes and start pulling and take us into metaphase, which is where we left off the last time I saw you. So the chromosomes are in the middle and they're being tugged on, and in anaphase the centrosomes each get their own way and they both tug those chromosomes apart. So to show this process happening, we need to start by drawing our cell. But our cell looks a little different right now in anaphase. The cell is actually, as it's pulling apart from each other, the cell starts to have this little cleave, this little indentation right here. And this is the first part of the cell separated into two parts. So we have our centrosomes on the north pole and centrosomes here on the south pole. And in the middle, we have our green chromosome, which just got yanked away from its other, since being pulled through the cytoplasm, it's being pulled in the upward direction. Then I'm going to draw it like that. And since the bottom one's going to get pulled in the downward direction, I'm going to draw it like this. But they just got pulled apart from each other. So we have the green ones separating and the purple ones will also be separating, and they're both being pulled towards the centrosomes. The centrosomes have those spindle fibers which are attached to these chromosomes, and so next we'll draw those. So these centrosomes have those attachment points to the chromosomes. They're also out here pushing on each other so that they can get their way and they can grab the chromosome that they all want. So they're attached there. And so it's kind of like a fishing reel. These centrosomes are reeling in these sister chromatids, these halves of the chromosomes. And so let's summarize over here on the side of what's happening here in anaphase. We'll say that the spindle fibers pull half of each chromosome, because it was a chromosome as they were attached, and so each side of the cell is going to be pulling half of each chromosome. So they're both an exact copy of each other. So if the north side gets a green half and the south side gets a green half, they both have the same information in them. So those spindle fibers pull half of each chromosome to each of the poles, to each pole. That happens in anaphase. And then we transition into the next step, step 5, which is called telophase. Kind of like telephone, but telophase. And in telophase, our cell has an even bigger cleave starting to form here. So I'm going to draw even more of a cleave happening here. And in this process, our centrosomes are still, we have one set on the north and one set on the south. And our chromosomes now are pulled apart from each other, and so they actually are going to start to decondense. So I'm going to start to draw them like this again, because they're not in that chromosome shape anymore. They're starting to go back into that chromatin state, but they're kind of in that in-between stage right now. But they're really vulnerable when they're in this stage. They're not protected. And so the cell knows that it needs to start to build that nuclear membrane around each set as it unravels. And so we're going to draw a dashed line here to show that the nuclear membrane is actually reforming. Alright, so we're going to say here that those spindle fibers that we've been talking about, the spindle fibers actually dissolve in this step. They're no longer needed because they've pulled the chromosomes apart from each other. The chromosomes are starting to decondense, and so the spindle fibers dissolve. The nuclear membrane, though, knows, or it senses as though the chromosomes need to be protected, so our nuclear membrane is going to reform. And then, as we were talking about, those chromosomes start to unravel. see, chromosomes start to unravel and turn back into that chromatin state. And then we transition into the final step, which is step number 6. And this is called the cytokinesis, because we're going to split the cytoplasm, so cytokinesis. And in this step, we now draw our cells, and then they're not separate and apart from each other, but there is a cell membrane in between each of them. And each of them now has a fully formed nuclear membrane in them, and all the other organelles too. But to keep it simple, we're just drawing the nuclear membrane. And inside of there, we now have the DNA has turned back into the chromatin state, and each side has an exact copy of the green, and each side has an exact copy of the purple, just as it wants to be, to live its happy life as it lives throughout the rest of its life in interphase, because interphase is what they're doing in between all of these phases. But here in cytokinesis, we see that the cytoplasm, which is the jelly stuff inside of the cell that takes up most of the space. It's like the air inside the cell. It's not air, it's actually a jelly-like liquid. But that cytoplasm is divided, which is now going to form, so cytoplasm is divided, forming 2 separate, but remember, identical cells. Which means then that each of those 2 cells are now going to go and start their lives. And when they start their quote-unquote life of doing their job, each cell is going to then enter interphase, but they're going to enter the first part, so that'd be G1. So each cell enters G1 of interphase. So if we look back at that pie chart here, once the cell has gone through mitosis here, it then starts living its life here in the G1 phase, and then it transitions into the S, where DNA is synthesized, and G2, where the rest of the organelles are synthesized, so that it then goes through mitosis, which is that small sliver of its life. So when we talk about mitosis, we're not talking about all of the life of a cell, but mitosis is several different steps. Mitosis involves prophase, prometaphase, metaphase, telophase. Metaphase is followed by anaphase, and then we have telophase. Now, as anaphase and telophase are happening, cytokinesis kind of begins somewhere in this time period here, because really it's whenever the cell membrane forms between the 2 cells, and that happens as those final steps are occurring, where the DNA is becoming protected by the nuclear membrane. So all of this is mitosis. The rest of a cell's life is spent in interphase, and that includes the G1, the S, and the G2 phase. So most of a cell's life is here in interphase, and that tiny little sliver there is mitosis. Now the cool thing is that with current technology, since we've been able to have access to microscopes, so it's not too current, but still in science, being able to look at mitosis through a microscope is awesome. And so we can actually take a look here. This one's not through a microscope. This is drawings that somebody made, a scientist made, after looking inside of a microscope. So you can see that, there's not just 6 phases here, because it doesn't happen in one step and then all of a sudden the next step happens. It's a constant transition. It's kind of like watching a dance. One move flows into the next move. And so as you can see here in A and B and C and even D, this is all the process of those the DNA condensing into the chromatin and then into the chromosomes. And then you can see here in E, we actually see that the spindle fibers are starting to attach to the different chromosomes. And in F and G, they get pulled into the middle. So both of those would kind of demonstrate metaphase. Then in H, we see those chromosomes being yanked apart and ripped apart from each other. And in I, and then we kind of see that the cells are starting to separate from each other. So it's not always like a 6-step process that happens 1, 2, 3, 4, 5, 6 in very specific ways that you're in step 3 and now you're in step 4. But we have to label each step so that as scientists we can say, oh, something happened in the metaphase that went wrong, or where did the thing go wrong at if something doesn't happen right. So it gives us a way that we can actually talk about this sliver of time called mitosis by calling out each of the different steps. Now, what we're going to be looking at is we're going to look at some newt cells. So some newt cells under a microscope going through mitosis. You can see here the chromosomes. You can hardly see them here, but they start to condense, and they're a little bit more visible here. But all of this black right here are the spindle fibers, and the centrosomes would be in this region, pulling on all the chromosomes, getting ready to yank them apart. They're here in the middle. They get yanked apart from each other, and then this is showing them what it would look like kind of in 3D. So let me get rid of my scribble. All right, if we look at it with some fluorescence, you can see the microtubules, or the spindle fibers, are green, and the chromosomes are going to be blue. So the chromosomes condense, and all those spindle fibers go out and grab a hold of those chromosomes as they're condensing. That'll get pulled here in the middle of the cell. So this is like a three-dimensional shape here. And then they get pulled apart from each other until finally we're about to start and have a new cell in between here. And now we would have our two different cells. So this one early cell in A then creates the two cells that are in H. So we have our one mother cell, and we end up with two daughter cells. And again here we have, this one is actually a kangaroo rat going through mitosis. And you can see a very similar process here. Chromosomes condense, meet in the middle, they get pulled apart, starting to make two cells. We have two cells. So that is the story of mitosis. These are the chapters from the beginning, or the introduction of it all, into the conclusion. We start with a mother cell and we end with two genetically identical daughter cells. And now you know the dance of mitosis. Let's go cut a rug.